Good evening, everyone. As Dean of the Faculty of Science, it is my pleasure and honor to welcome all of you to the inaugural lecture of Professor Sophie von der Heyden. So I would like to extend a special word of welcome to two members of the senior management of the university joining us this evening, Dr. Ronel Retief, our registrar, and Professor Sibusiso Moyo, Deputy Vice-Chancellor, Research, Innovation and Postgraduate Studies. I would also like to welcome Professor Guy Mitchley, the Interim Director of the School for Climate Studies, our Vice Deans, Professor Frikkie Scholes and Ingrid Revitsky, and the Head of the Department of Botany and Zoology, Professor Teresa Vosler. A warm welcome also to family, friends and colleagues from Stellenbosch University and other institutions. Those of you who are here in person as well as those joining us online. Finally, also please allow me to extend a very special word of welcome to some very important guests this evening. Professor Sophie von der Heyden's husband, Constantine, their children, Helene, Daniel and Anna, her parents, Birgit and Eric, joining us online, and her brother Jörg and his family. An inaugural lecture is a real highlight on our academic calendar, and we are really honored that so many of you are here this evening to celebrate this special milestone in Professor von der Heyden's academic journey with us. I'm now going to ask Professor Guy Mitchley to introduce Professor von der Heyden.
It's really my great pleasure and privilege to add my words of introduction to this inaugural lecture by Professor von der Heyden, who I will now refer to as Sophie from now on. Yes. It is indeed a very special event, as Louise has said, an incredibly important milestone in the career of an academic. So this is a great moment. I'm also delighted that Sophie will be formally associated with the School for Climate Studies, and where I serve as acting director, as Louise said, in addition to her primary affiliation with the Faculty of Science. And I look forward to her contributions to the further development of the school. This inaugural event is a celebration of a working lifetime of scientific scholarship and achievement by Sophie, and it's a well-deserved recognition of an impressive scientific career that has achieved much and will achieve much more. Sophie's academic career is exceptional. She's converted a strong undergraduate and postgraduate track record, including such institutions as King's College in Oxford, much revered here in the Southern Hemisphere, into an already powerful contribution at Stellenbosch University. So we're extremely lucky to have such an excellent scientist among our staff here in Stellenbosch in South Africa, and indeed on the African continent. My introduction comes from the perspective of a colleague, collaborator, and a co-supervisor of some students, who's now worked with Sophie for almost 10 years, and has had the experience of being coordinated by Sophie in various academic enterprises, including the Global Change course. Sophie's very well organized. She's very well organized, and she also has the gift of managing a very good work-life balance. So she manages her jobs perfectly, and also her family. So that's quite an amazing achievement. I've also been generously included in some of her funding proposals, not all, but some. And she has a very high success rate, and that work is producing students, first-rate science, excellent students, very well-prepared students, and at undergrad and postgraduate levels that are going to really help South Africa into our challenging future that faces all of us. Speaking of her students, she is very well respected and appreciated by her students for her attitude, her positive attitude. She's a can-do, matter-of-fact scientist, and she has an irrepressible drive to do amazing things very well. And you can see that in her publication list and in the excellence of the students that she has graduated, of which there are many, I think something like 60. On the other hand, she's also very approachable. She's supportive of her students. And she's a problem solver in a profession that is beset by problems. She gets over them. And she's a big thinker in a field where so many are mired in detail and obsessed by triviality. So Sophie does see the big picture. And we're going to see that this evening in the questions and issues that she's tackling, which are right at the forefront in the intersection of some novel methods, techniques, and an ecosystem, the coastal zone, which is at the forefront of impact from climate change and other human impacts. So I think we can all look forward to a thoroughly engaging lecture and take us on your journey. Thank you.
Thank you, Guy. Thank you, Louise, for my introduction. Apparently I have 45 minutes. So, as you all know, I love talking. I might be a little bit over. And I think one reason why I'm going to be a little bit over, because I'm going to start today actually by thanking a whole bunch of you. And it's the one time that I cannot talk impromptu. You know, if you've been to my lectures, I can put up one photograph and talk for 45 minutes. But I had to, this is the really important stuff. And I kind of just want to say, this is my most important lecture that I've ever given. You know, I've spoken as keynote speakers at big international conferences, obviously my everyday lecturing. I do a lot of international work, but this is really the one that counts most for me. So, yeah, which is why I need a prompt note just for my thank yous. So tonight is, you know, you were invited on to a celebration of a remarkable academic journey, and I've got the gown to prove it. There we go. But even more so for me personally, it's a real celebration of my community, of my family, my friends, my colleagues, everyone that is here tonight, and those also further afield. So it is a night of acknowledgements and a thank you, because especially for my family and friends, because I'm actually here because of you. You know, you've all played an incredible role in laughter and support, and just you're always just there, and it's amazing. So it's really my celebration of you, hopefully, and there's lots of wine afterwards. So you're all here in your own special way as part of my academic journey. I do have to kind of pick out a few people, and obviously the first one would be my absolute rock, Constantine, my three minor rocks, Helene and Dan and Anna, and my parents, Birgit and Eric, who unfortunately can't be here today, but I really hope, Mom and Dad, that you can see me online and that you're able to hear and you've got your hearing aids in. And mein Bruder Jörgi und seine Familie in Deutschland, whose English isn't brilliant, but I'm hoping that they'll follow this. And my wonderful in-laws and my extended family, who even sent me a few messages to say, what is the dress code? So thanks everyone. And then my brilliant friends and partners and parties. I mean, you know who you are. There's the rest of you all here. So thank you for being here. And then thank you also to my colleagues, both in the department and at the Faculty of Science, you know, who've really been an incredibly supportive network. It takes a lot to come to this position, you know, to get here. But if it wasn't for the incredible admin staff and for my colleagues, you know, and for the thoughts and the coffees and the problem solving, like I mentioned, because it is a bit of a roller coaster sometimes. This evening is also for you. And then to my students, there's quite a few here. So nice to see you all. actually probably made me the most nervous, because all of you give such brilliant talks, and I'm forever saying, but look at the detail, and think about structure. So not a word, okay? After tonight, this is zip. And then finally, I would just like to thank Jonathan Dale and the team that put this evening together, and Suzette at the faculty, that we can all be here together, and hopefully you'll understand some of what I'm about to tell you, which is kind of what I do when I'm not partying and spending time with my family and doing all the stuff. But really, thank you everyone. It's such a privilege to be here and to share this with you this evening. Okay. All right, so I start my formal academic lecture with a little bit of a confession, because it's actually been more than 15 years. But you remember, you never ask a lady her age, and you probably shouldn't ask a professor how long you've been doing this. So it's more like 18 years, but 15 is a nicer number. But really what I wanted to provide to you today is just an overview of the work that I started when I arrived here in 2005 to where we are now. And really, to begin with, credit where credit is due. And these are hopefully all my students that I've supervised in time, as well as some really incredible postdoctoral fellows, on which kind of this research is built. I come up with the ideas, yes, I look for funding, but the actual research is predominantly carried out by this incredible group of people here. So thank you. This is kind of I'm here again because of you. And obviously my amazing collaborators and funders, both in South Africa, but also globally. So my story and my affiliation with Stellenbosch University starts in 2000, well, it starts a bit earlier because that's when I met my husband overseas. But it kind of took me from Northern Europe, from Germany and England, where I studied, to wine country down here in Stellenbosch. It was an easy move. I was not dragged here kicking and screaming, so it was a great move. But this was in 2005. I finished my PhD at the University of Oxford, and then it became well, what now? And we made the decision to move here, and I was very gratefully accepted by Conrad, Mattia, and Rory Barry at the time. Rory's now in the US, but really they were the foundation of why I'm here. And it's actually such an incredible place to be working in as a marine biologist, because what this shows you here, this is a NASA kind of remote sensing video, but essentially what it shows you is that the oceanography, so the ocean currents around Southern Africa, are incredibly dynamic. But I think you'll all see, and I'm pointing here at the ocean currents, is that on the east coast there's a warmer current, the Agulhas current. On the west coast is the Benguela current, which is kind of which is a lot colder compared to the Agulhas current, and there's lots of upwelling. As we all know, at Cape Town in summer it gets pretty cold. But essentially it makes this place, in Southern Africa, a really incredible place to be a marine biologist, because we have this entire playground of incredible marine biodiversity that's kind of, that's driven by these really quite amazing boundary currents that you find nowhere else. So what this map shows you, and this is just the introduction to kind of set the scene, but what you see in front of you is basically from the National Biodiversity Assessment from 2018, and all these different colors along the coastline and also offshore basically show you different kind of habitat types. So that means there's a lot of different habitats for marine species to live in. And again, that makes it really very incredible to be working in the system. So, you've all hopefully been to the beach quite a lot. In fact, I know most of you have been to the beach quite a lot because we've been there. But, some of you even have the privilege of spending time with us traipsing around seagrasses. But there's an incredible diversity of marine ecosystems, ranging from rocky shores and mangroves and sandy beaches to seagrass, to these beautiful underwater kelp forests, some coral reefs on the east coast. Oh, look, more seagrass. And basically, there's this tremendous diversity of habitats and of marine life. And that's mirrored in where different species are found. So basically what this shows you is that around the coastline of South Africa, okay, what you can see here at the bottom is basically 100-kilometer units. Along here, this is the number of species. And what you can see is that from the west coast, which is this part here, going up to the south coast, you have an increase in species richness. So basically, species richness is just the number of species you find in a particular area. Okay, and I want you to remember this graph for later. So in South Africa, we also have really interesting ways of how species are distributed. Okay, are you still following? It's not excellent. Okay. But what's really interesting with all of this is that a lot of species, and these are just random species, they might not even be South African, okay, I just found them on Google, a lot of the species actually have distributions that span this incredible environmental gradient. So you can find populations of crabs and fish and mollusks, but not necessarily these ones, that live on the colder west coast. But you'll also find them kind of ranging into the warmer east coast. So that, as a biologist, is fascinating. And it brings up all these questions of, well, are these guys able to interbreed? No? If they're not, what prevents them? Why are they there? It's essentially what we're still trying to figure out. But Southern Africa is also one of the global marine hotspots for ocean, for changes in ocean temperatures. So this was a paper published in 2013, 2015 that kind of highlights down here, number 17, that Southern Africa is really one of the kind of sentinels for a changing marine system. Okay? So again, as a biologist who's interested in climate change and how species respond to climate change, this is a really cool place to work. And then to add to that is that some species are shifting their ranges in response to this climate change. So we've got, for example, documented range shifts of fishes from tropical waters into more warm-temperate waters because the warm-temperate waters are getting warmer. And now we've got situations where we have entirely novel communities. So we've got species that are moving into areas where they never lived before. So what does that mean for communities? And more importantly, how, as marine biologists or as marine scientists, are we able to figure out who's moving and who isn't? Because it's actually quite difficult to kind of track marine species, right? Many are mobile. How do you follow a fish larva? Barely follow a shark. So those are a lot of the kind of problems that I work on. So I want to kind of break this down. I did promise you 15 or 18 years of molecular ecology. And I think this is such a nice way of illustrating my progress, because here's young Daniel, dressed in pink, he's a baby, so lying on a beach in Betty's Bay, and this kind of starts back in 2005. And some of those very early questions go back to understanding the basic kind of the basic biology of where species are in space and in time. So how do those ocean currents, how do they promote or prevent species interbreeding between warm and cold water temperatures, for example? If we look at enough species together, do we find patterns that are shared between them? Or do we find that species respond in the same or in different ways to the environment? And then kind of depending on the molecular ecology, and I'll talk to you a little bit about kind of genetic markers just now, but how do we monitor marine communities in a changing climate? To what extent does the environment shape these marine populations? So there's really been an evolution of the kind of questions we've been able to ask. Also, because some of the techniques that we were able to use in the mid-2000s compared to now have fundamentally changed the way that we're able to answer these questions. I will test you on this all later. Okay, so take good note. Basically, what I'm trying to get across with this slide here is that our ability to answer the questions about who lives where has really changed depending on the kind of techniques the genetic techniques that we've been able to use. So in 2005, we were really constrained to using single mitochondrial markers. Okay, but basically what I just want you to know, it's a single marker. It gives you kind of one gene sequence for one individual. Okay, and we did that for about 30-odd species, and I'm going to show you those the data just now. What's been really fascinating and extensive, and as Romina knows, a real roller coaster of analyses. But since about 2015, 2016, we've been able to extend our molecular toolkit to look at genome-wide markers. So basically, instead of just looking at single shortest gene sequences, we can now randomly sequence bits of a genome, and that gives us a lot more data to answer our questions. So we've gone from very little data to a lot of data, which is also computationally quite demanding and has some challenges, but it's been great fun. And why marine systems in the first place? So what this graph here shows you is basically the IUCN category, which is basically a category of how endangered how endangered a species is, between terrestrial species and marine species. And what you can see, those grey bars for marine species basically show you is that a lot of marine species in the oceans are not reviewed, or they are DD data deficient. So it basically says that we know so little about marine ecosystems and the species that live there compared to terrestrial species. So, and as a scientist, if you're able to spot a good gap, you've got to grab it, and this is a huge gap, okay, as my students know. And now we'd think, and really what's kind of brought all my research together is conservation planning and decision making. But how do you put into place a protected area when you're working with species that move? Okay, not move like fishes, but move because they have larval stages. And we work in these incredibly dynamic oceanographic currents. Those larvae get transported. So how do you decide where to put a marine protected area when everything that you're trying to protect could potentially move? So those are the kind of questions, again, that I've been grappling with in 15 years' time, in the last 15, 18 years. And our research, has really had two major drivers. The first one, which is what I started, was sequencing thousands and thousands of hake, and then Romina carried on sequencing thousands of hake, and now Courtney sequencing thousands of anchovies and kinglet and hake, and we never got to eat them. So we did a lot of work in offshore ecosystems, but primarily for commercially exploited fishes. So I'm not going to talk to you about that. I'm really going to focus on coastal ecosystems, for which we have a lot of data, and it's and I love a good rock pool, as everybody knows. So I'm going to just break it down a little bit and take you from kind of a single taxon population genetic approach to what happens when we look at multiple species, just as an interest. So this little dude, Tylus granulatus, is actually an isopod. It's a very big isopod. It's about 2 to 3 centimeters big. You'll only find it on the west coast, and it's actually gone extinct in places like Bloberg and Table View, all those sandy beaches where people live, because people do bad things to beaches. But essentially what this shows you is, just as an example, population genetic study where each of these colors is a different population that we went and sampled, we extracted DNA, we sequenced this particular species, this little isopod. And what we then do is we kind of look at how related the individuals that we sampled along this coastline are. And by using something as simple as a haplotype network, we can kind of look at the relatedness. So each of these different colors are individuals from different populations. And what you can see is that a lot of the colors are not shared between populations, right? So here we've got all green, here we've got all purple, here's a little bit of mixing. And what this shows you is that these little isopods that we sampled at each of these different beaches are in fact genetically quite different to each other. And why is that important? Well, it means that they're not able to interbreed very easily. So if you lose a population, it's very likely that that population is extinct. It's got a unique genetic signal that you'll never have again. Okay? And this is really a story about losing evolutionary potential, because we've already lost some populations of this particular isopod. So if you upscale this, here is one species. Now you put this into a big boiling pot of data analysis, and you add 30 species. And that then begins to, very powerfully be able to show you patterns of things such as genetic diversity, which is very important. The more genetically diverse a population is, the more resilient it is potentially to climate change or things that people do to marine species. So I don't need you to remember this map, but basically what it shows you is that there are hotspots of genetic diversity. So everywhere that is greenish or yellow, here the southwest coast, here the south coast, has elevated levels of genetic diversity. And that's important because that is where we have this evolutionary potential that can allow species to persist in this very uncertain future. Okay. And what's very nice is that you can then look at things such as ecologically or biologically significant areas. In this case, this is the Walker Bay EPSA, and you can actually see that a biological signal very nicely is overlaid with a genetic signal. So we're beginning to find these patterns of high species diversity, but also high genetic diversity. And from this, you then have really, really good data to kind of say, well, from a conservation perspective, these areas are very important. So science sometimes is a little, not sometimes, everyone here in the front row, science is often luck, right? It's being in the right place at the right time. And in 2015, I had a very clever student, Nikki Fair, who said she would like to work on something endangered. I said, oh, that's cool. I read something about seagrass. That's endangered. Let's do a seagrass study. We managed to get some funding, and that has snowballed. I'll hopefully be able to finish off my lecture with that. But Nikki's study was the very first in South Africa that used not only single markers to look at the genetic diversity of seagrass populations, but that used that genome-wide approach. So where we had thousands and thousands of markers to look at how seagrass, in this instance, seagrass is distributed around South Africa. So that was about 10 years in the making. Nani, this is you. Okay. Now, what did Nikki's work show? Well, Nikki's work was super interesting because it was really one of the first examples in Africa where we took this enormous data set of genetic markers and we could tease apart different facets of it. So there are markers that are under selection. So for those of you who can't remember Genetics 101, basically markers under selection are those that are responding to changes in the environment, and then there are neutral markers. So we don't worry about those too much. Well, we do, but not in this case. But what Nikki showed was that with just 3 different markers under selection, we were able to show that there are in fact 2 big lineages of seagrass in South Africa, and these are kind of shown by either a very kind of big spike here on the graph or a very low one. But importantly, it really gave us this new tool and this new kind of insight into how to work with these kind of thousands of different genetic markers. So what you can see here is basically all these different colors are different lineages and different populations. But importantly, what Nikki also showed was that she did some really interesting modeling where she showed that in places that have really high human impact through, for example, habitat destruction or fishing, the genetic diversity of seagrasses was lower. So that gave us a really direct link that people not only impact marine species randomly, that we're also beginning to erode their ability to respond to climate change in the future. And that's really bad, because this evolutionary diversity has kind of amassed over tens of and hundreds of thousands of years. You know, in the space of a few years, humans are very good at eroding that. So, but again, very important data to give to conservation planners to kind of say we need to be a lot more proactive in conserving genetic diversity too. We can then map that. I'm not going to spend too much time, but it's just an example of some of our work where we can map the genetic diversity signal onto different habitat types, critically endangered, endangered, to least concern, and kind of say, well, where are there areas of high genetic diversity that fall into critically endangered places? Because then we can say that is where you need to concentrate your conservation efforts. So I've given you some examples of patterns, right? I've said there's high genetic diversity, there's low genetic diversity, but why are those patterns there in the first place? And I'm going to take you back 22,000 years, which is really the peak of the last ice age. I know you've all seen Ice Age, it wasn't like that. But 22,000 years ago, you know, Southern Africa was a very different place, because at the peak of the last glacial maximum, sea levels were about 120 meters lower. So you could be at Cape Agulhas, where it is now, and then you could still walk 100 odd kilometers until you reach the tip of South Africa. So a lot more land. But also what that did, that fundamentally changed what the coastline looked like in that time. So in 2014, Jessica Thomas did a very nice honor study where we noticed, and I hope you can see that, but there are green bars and pink bars. Okay, those are basically two different genetic lineages of this particular little fish. And if you look at their distribution, the pink bars are always here, the green ones are always here. So what Jessica did in the computer, she dropped the sea levels by 120 meters. And we looked at where there were rocky places. And basically, we showed that that 22,000 years ago, there were two major rocky shore areas left in South Africa where we can be certain that there was rock. In the southwest coast, remember all that high genetic diversity? This was probably where a lot of species managed to survive, that drop in sea level. And on the east coast here, around Cape St. Francis, which I know is now very popular for teenagers, New Year's. But importantly, you know, what we were able to show here was that a process 22,000 years ago, 22,000 plus years ago, has shaped the genetic diversity of species that live today. Okay, so it's not just about things that we do today. A lot of these patterns are very, very old. It's the same for seagrass. We kind of showed that one of the big refugial areas in the last glacial maximum around then was the southwest coast, where we find a lot of elevated genetic diversity. So lots of different bits of information coming together. So that was a historical angle. Remember that incredible photo that I showed you earlier on of these very diverse ocean currents? Pretty much all of you, I think, will agree that, well, what about temperature? What about salinity? Surely if you've got populations that live in a cold environment and you've got populations that live in this warm environment, maybe they're adapted to those environments. And you would be 100% correct. So if you just look at this kind of tree thing at the bottom here, which is worked by Peter Teskey, basically all of those, this is genetic data for goby species. All those different colors, or these 4 different colors, are genetic signals from a very cold population, a cool temperate, warm temperate, and subtropical. So this shows you that temperature is a really, really important driver of where marine species live along the coastline of South Africa. And it's the same for, this is Erica Nielsen's work. She looked at 3 different invertebrate species. She showed that temperature was very important for one, but also salinity. But basically the take-home message is that there's really strong environmental drivers that shape populations of marine species in South Africa. And this is just another data set from lots and lots of, again, thousands of genetic markers that kind of show you these outlier loci that are probably under selection, and they come up all the time for each of the populations in comparison to those neutral loci. So the environment is critical for shaping where marine species are in space and time. Okay. And I'm wondering, I'll go back to my question here. So a lot of the data that we have, particularly kind of for low-income and middle-income countries, for natural populations, still come from single marker studies. Okay, but there's a big drive globally to now focus on, on producing these kind of really big-scale, very wide, thousands of marker data sets, which are expensive, and it's really difficult sometimes working in an environment where the technology is being driven by the Northern Hemisphere, okay? And the capacity is there, the money is there, the people are there. And we're trying, we're still trying to catch up. So that's been, but that's really made me think about the value of these single marker data sets, particularly for conservation planning, because sometimes that's all you've got. So, my PhD supervisor, Tom Cavalier-Smith, always said some bread is better than no bread. And I told even if the bread's not quite fresh, at least it's usable. And that's kind of how I've always looked at this problem. So with Erica Nielsen, again, she was really interested in how we take genetic data and how we use it for conservation planning. But she ran a whole bunch of different models to really try and understand the value of genetic data for conservation. So what we've got here on this map, each of these little stars is a different place where we sampled a few species, and then this is a kind of conservation prioritization carried out just on habitat data. Okay, so basically every one of these little cells is a particular space along the west coast. If it is in dark blue, it basically has been selected a lot, okay, for habitat, for example. But really importantly, we were one of the first labs in the world to kind of show that if you add genetic data to something like just to a conservation plan based on just habitat or species diversity, the conservation outcome changes fundamentally. And that's really important because genetic diversity is not often included in conservation, but it is essential, right? So that was super cool. We've done so much, we've done really, really great research in this area, and we've really shaped our understanding of the importance of genetic data, not just for South African marine species, but for kind of conservation science globally. And we did some very nice studies that kind of showed, basically, again, I want you just to kind of focus on, how big these bars are here. But we basically showed that if you have enough species even just for a single genetic marker, they are able to capture as much conservation kind of area as if you have these data sets with thousands of genetic markers. And that's really important because it means that we're able to utilize these older single marker genetic data sets that are really prominent, particularly for African conservation, and we can really get good data out of them. And that's very important if you have no money. Guys, we still have lots for C-class research. Okay. So I'm going to spend the last few minutes, because it's really a whirlwind tour of a few years, to kind of taking you back to this idea of, we live in a really changing environment. We hear it on the news. There's wildfires. There's really a lot of torrential rain and floods. I mean, we've seen it in the space of 7-8 years. We've gone from a drought to the Yser River breaking its back. So we're living in this environment of global change. And I showed you earlier on that some species actually change their distributions. They're looking for places that are nicer to live, right? So as the East Coast is getting warmer, species are probably moving down to where it's cooler. South Africa is a bit strange because everywhere else pretty much the ocean's getting warmer. In South Africa they're getting warmer and cooler. What we're seeing is we've kind of intensifying southeaster winds. We've got much more intensive upwelling in summer. So we've got colder water that we used to just have on the west coast coming down around the southwest coast. And with that, there are species that are shifting their ranges there too. So kelp forests, for example, used to kind of end kind of around Bailey's Bay. You can now find them in the Guip, and they're extending further eastwards. So there's almost a squeeze of warmer oceans on the east coast and cooler oceans on the west coast. Not good news if you're an endemic south coast species. Okay, but how do you begin to figure out who lives where? Species are shifting, and it's, like I said earlier on, it's difficult to track a marine organism. Okay, yes, you can put a marker on a fish, but what about snails? What about, I don't know, give me some marine species, crabs. It's difficult, especially because so many marine species are broadcast spawners. So, they've got larvae that get washed around these oceans. So a few years ago, we started using a technique called environmental DNA metabarcoding. And some of you have heard me talk about this late at night, but essentially is that you sequence the species diversity in our case, a liter of water. So we go out there, we collect at all these places. I've got some very lucky students who have gone to every single one of these triangles. It's like a 4 to 6 week field trip. 2 different seasons, so it's always fun to be a student in the Heyden lab. But basically they essentially collected water, they filtered it, and then we sequenced to see what is in there. Even at midnight during load shedding. Okay. What this shows you is out of about 100 litres of water that we collected around the entire coastline of South Africa, is that we were able to detect 115 of the approximately 200 families of marine fishes. Okay, so without seeing the fish, we were able to kind of say, well, look, we've got this DNA signal that comes out of this liter or 3 litres of water, and we're able to kind of, develop a tool where we're able to look at where species are. Again, in space, so where along the coastline, and in time. And that is incredibly powerful because, like I said earlier on, it's difficult to figure out where everything lives, especially where it's shifting. So with environmental DNA metabarcoding, we now have developed this really novel, this really great technique that just by filtering water, we can figure out where species are. This is just another, a kind of chart. These are all our different sampling points, but basically each of these colors, and some of the orange is very similar, but there are only so many oranges you can actually have in R. But basically, each of these little blocks is a different family. So now you can see, for example, here at Port Nolloth, very few species compared to some of the other places with lots of species. Okay, so very kind of hyper diverse ecosystems that we can sequence, literally just from a few liters of water. Remember, I said please keep in your head that species diversity graph at the beginning. Well, here it is again. This is for rocky shore species. But now, if you look at environmental DNA sequencing data for fishes, you can see that we have exactly the same signals. We've got low diversity on the west coast, and as we go up the east coast, a lot higher diversity. So we also know that our technique is powerful enough to detect some of those different patterns in species diversity. I'm so on time. It's so great. I thought I was going to be hours over. But I can't really leave here without talking a little bit about seagrass, because this is a very important part for anyone, certainly for my students, but I think for all of us in life. And that's kind of being adaptable and taking an opportunity when it comes. So we kind of went from, scrabbling around looking for genetic diversity signals of seagrass to building something that is really quite all-encompassing. So my students at the moment are really involved and are teaching me huge amounts actually about things such as how seagrasses respond to different lights, to how they respond to thermal stress. We're sequencing the bacterial communities that are associated with seagrasses that can underpin things such as carbon cycling and even seagrass health. I've had students working on pollutants. I mean, what do I know about pollutants? I know a lot more now than I did a few years ago. Micropropagation with NOx. We're kind of figuring out how we can grow seagrasses from incredibly tiny bits of plant so that we can restore it back into populations where seagrasses have been lost, and that's actually happening in South Africa. It's happening globally, and it's a big cause for concern. And we're venturing into things such as blue carbon and thinking about carbon trading. So incredibly diverse, and really I think a great illustration as to how important it is to kind of, as a scientist, as a mom, as a friend, to be adaptable and to kind of walk this incredibly exciting journey. And with that, I obviously have to acknowledge my funders that are wide and diverse in South Africa and globally. But I want to thank all of you, like I said, for being here. And then really, some of the happy moments from the lab. Enjoy.
Thank you so much. It was super interesting, even from somebody who's not a scientist. It often happens that when you are in a managerial role, you have to speak on topics or in spaces that take you out of your comfort zone, and this is one such an occasion. In my academic field, arts and social sciences, the ocean, generally speaking, evokes a human response. People often express strong emotion when they refer to the ocean. Many poems and songs have been written to either marvel at the beauty of the water mass or to lament the separation that it brings. If you think of My Bonnie Lies Over the Ocean. Tonight's lecture prompted us all to look beyond the surface and to marvel yet again, but this time at the wonder of the marine populations along the southern African shoreline, and at the way molecular data can be utilized for the conservation and management of marine resources. It's also fascinating that Prof. van den Heyden's work reaches far beyond the field of aquatic biology, which was a focus when she first embarked on her academic journey as an undergrad student. She exposed us to fascinating insights about the evolutionary dynamics of one of the world's most unique ecosystems. And we also learned about the relevance of this work for the expansion of molecular approaches for studying natural systems and the applicability of that for conservation generally. It would be remiss of me if I did not recognize that on the eve of National Women's Day, we are celebrating a woman in science this evening. A significant gender gap has persisted throughout the years at all levels of science, technology, engineering, and mathematics, the STEM disciplines, all over the world. Even though women have made tremendous progress towards increasing their participation in higher education, they are still underrepresented in these fields. Gender equality has always been a core issue for the United Nations. On 20 December 2013, the General Assembly adopted a resolution on science, technology and innovation for development, in which it recognized that full and equal access to and participation in science, technology and innovation for women and girls of all ages is imperative for achieving gender equality and the empowerment of women and girls. But gender equality and the empowerment of women and girls will also make a crucial contribution not only to economic development of the world, but to progress across all the goals and targets of the 2030 Agenda for Sustainable Development. So it works both ways. Prof. Heyden, you have been recognized in many ways by national and international bodies, as can be seen from your CV. And tonight we are honored to celebrate your inauguration as professor, which is the university's way of acknowledging your vast contribution to your discipline and beyond. Congratulations for this achievement.
Thank you.
Good evening, everyone. Sophie, what did you do? Did you send out a tweet or something?
No, they are all my friends and family.
No, I heard, because there seems to be a lot of partying happening.
Not with us, we're clean living.
Thank you for a wonderful talk. It was fascinating, and I'm going to, actually, I think of Sophie as Professor Seagrass. Actually. So Sophie is actually one of those staff members of mine that has really diversified her research career. So when eDNA started and it became something that no one in South Africa was really doing, Sophie took the bull by the horns and started an eDNA lab. And then all of a sudden, she really set up this huge seagrass experiments and research into seagrass because she saw how important and the gap in the research and the value that seagrass has had in conservation and in the climate change spheres. So Sophie has been very forward-thinking and quite innovative in the research that she has followed in the last 15 years. But as a staff member, she is a very wonderful person to have in the team because she's a great team player. So if you really want something done, which I've now realized, she's going to be my new marketing manager. Because I can just see how she can get people to follow and do. So she will always be there. She will always be prepared to do something. And as Guy was saying, very efficiently and very coordinated and very quickly. So it is great having you in the department. And you have really been very supportive, and I'm very glad to see you at an inaugural lecture. I'd also like to thank Jonathan Dale and his team for putting this all together and having everything run smoothly. Thank you very much. And of course, I'd like to thank Sophie's family, all her friends, everybody that has come out and listened to this inaugural lecture and celebrated this with Sophie and all of us. And I would like to invite you all to join us in the foyer for a glass of wine and some snacks. Thank you very much.